So almost all short reads based sequencing at the VIB nucleomics core is done with the AVITI sequencing system nowadays. Illumina sequencing is still used, but mainly for sequencing single-cell libraries. The AVITI sequencing system was developed by a company called Element Biosciences and is based on what is called the AVITI sequencing chemistry. We'll come back on that later. Just like in the Illumina system, sequencing is done on a flow cell, but the flow cell looks a bit different. The flow cell is unpatterned, so it's a glass slide, and it contains two physically separated lanes. And the range of the camera divides each lane into multiple tiles, just like in the Illumina system. The library preparation is very similar to the Illumina library preparation, but there's one additional step. So you make sure that you generate fragments in a certain size range, and to these fragments you are going to ligate adapter sequences in such a way that you get double-stranded fragments. So the original fragments that come from your DNA or your RNA are here in gray. So you have double-stranded fragments that are coupled to two different adapter sequences. And these adapters are typically Illumina adapters. That means that you can do AVITI sequencing on Illumina libraries. Then you're going to take your Illumina library, you're going to subject it to a denaturation step, so you get single-stranded fragments, and to these single-stranded fragments you're going to add extra adapter sequences. So these AVITI adapters, they are 48 bases long and they are added to the two ends of your fragments, and they will allow circularization of your fragments. So instead of having single-stranded linear fragments, you get single-stranded circular fragments. So Illumina adapters are supported. The adapters that you see here are the regular Illumina adapters. However, if you want, you can use AVITI adapters here in this step instead of the Illumina adapter sequences. But the VIB nucleomics core always uses Illumina adapters here because it will allow them to decide after the library preparation which sequencer they are going to use. You can work on an Illumina sequencer because you have an Illumina library, or you can work on an AVITI sequencer because you can use this library to add these AVITI adapters. So after circularization, you put your library onto the flow cell. The flow cell is coated with short primers that will bind to the adapter sequences and a chemical that promotes the attachment of the fragments to the surface of the flow cell. So in this way, your fragments, who are still circular, are captured on the surface of the flow cell. Now, and just like in the Illumina system, you need to have enough material to detect the sequencing signal. So on the surface of the flow cell, the fragments that are still circular are multiplied to form clusters of identical copies. And these clusters are called polonies. So here you can see them. So you have clusters on the surface of the flow cell. So how is this amplification step done? It is done without doing any PCR. So Illumina will do this amplification with PCR, but that generates PCR copies and biases. So in the AVITI system, they don't use PCR, but they use a system that's called rolling circle amplification. So how does it work? Well, you have your flow cell. Your single-stranded circular fragments are attached to the surface of the flow cell. And you're going to add a second primer. And this green sequence that you see here represents this second primer. So this primer is not the same primer as the primer that is coated on the surface of the flow cells and that is used to attach the fragments to the surface of the flow cell. No, it's a different primer, but it will also bind to a specific sequence in the adapter. And then they use a DNA polymerase, and that DNA polymerase will use this primer as a starter to produce a copy of the fragment. And because the fragment is circular, the end is never reached. So the polymerase keeps on going and producing copies until you have a very long single-stranded product with multiple copies of the original fragment. And these long single-stranded products are called polonies. So these polonies are very similar to the clusters in the Illumina system. The big difference is that they are not formed by PCR. Then the actual sequencing can start. So this is the surface of your flow cell. And here you have the polony, so the cluster that is attached to the surface of the flow cell. So a sequencing primer is added, and the sequencing primers are shown here in red. Your sequencing primers will bind to the corresponding sequence in the adapter sequences, but because you have multiple copies in one polony, it means that multiple primers will find an adapter sequence and will bind to the polony. So the polymerases are added. That's the gray-blue circles that you see here on the slide. So they will occupy the place where the sequencing primer was bound to the adapter, and they will add a nucleotide to the DNA strand that is being formed. So, and nucleotides are added in the form of avidites, consisting of 4 identical nucleotides bound to a fluorescent particle, as you can see here. And so for each base, the wavelength of the light that is emitted by the fluorescent particle is different. And this allows you to make a distinction between the 4 bases. So one base is added. The nucleotide also contains a chemical block that prevents a second nucleotide from being incorporated. So similar to the sequencing in the Illumina system. So we add the avidites. In each polony, one base is added to every copy of the DNA strands that are being formed in that polony. The excess of avidites is washed away. A laser is shown on the surface of the flow cell to activate the fluorescent labels. The fluorescent labels will emit light of a certain wavelength. That light is detected by the camera. The avidites are removed, the block is removed, and the next cycle of sequencing is initiated. So you start all over again. In each of these locations where a sequencing primer was bound, a second nucleotide will be incorporated, and after washing, the bases of this second nucleotide will be detected. The blocks are being removed, and we start over again in a third round of sequencing, and so on. So the sequencing is very similar to what we do in Illumina sequencing. But there are differences. So you have these avidites where you have multiple bases that are coupled to one fluorescent particle, and you have these polonies instead of clusters on the surface of your flow cell. So it's similar but different from Illumina sequencing, and the difference makes that in this system you need less reagents, it's more efficient, and less reagents means also that you have lower costs. So it's cheaper than Illumina sequencing. And additionally, it generates a less background signal.